All right, guys, let's take a look at gene regulation and expression. So how do we turn genes on? How do we turn genes off? And expression, and what is the result of that? So we're going to look at prokaryotic genes and eukaryotic genes. And prokaryotic genes, prokaryotes, of course, are organisms that do not have a nucleus, single cell organisms. Okay, so we learn a lot by looking at single cell organisms on how they work, and then we compare that to how eukaryotic or the true cells work, describing how genes are regulated in eukaryotic cells, so cells with nucleuses and us. And then look at what controls the development of cells and tissues in multicellular organisms, and so what's in control of the differentiation of cells that ultimately end up with that being us. So at first, let's take a look at prokaryotic gene regulation. So in prokaryote, there's some DNA binding proteins that regulate genes by controlling transcription. And those are specific proteins that attach to a segment of the DNA of a prokaryote that either turns on or turns off the ability for that organism to run that gene, replicate, make the protein or whatever they need. So one of the keys to that transcription in bacteria is the organization of genes into what they're calling operons. And so bacteria have about 4,288 genes, typical E. coli, and they will group those genes into an operon. And those operons have functions. And so the lac operon is a well-studied operon in bacteria that codes for lactose. So the lac operon is only operating when lactose is present. And when lactose is not present, the lac genes are turned off by these regulatory proteins that bind to DNA and bind and block transcription. So when this regulatory protein binds to the DNA, the transcription can't run. So yesterday, and come back to this as many times as you need, I showed you a video of that transcription from DNA to RNA, and then that RNA is expressed. Okay. So the way that works is there are some different areas, and so located this operon, these lac genes here is the code for lactose, or the proteins that are going to break down lactose into simple sugars. Okay, so we have this operator, promoter area, and a lac repressor gene. And then this is an active repressor gene. So we have a promoter and an operator, okay? And the RNA polymerase will bind here. So we know that RNA polymerase is what makes that copy of the DNA. The operator, whether it turns it on or off, that is going to reside here. So if the operator, if this repressor protein is in effect, it's going to stop that from working. And that's where the lac genes are, and those genes are the ones that produce the lactose. Okay, so when that lac repressor molecule binds to the O region, the operon region, that RNA polymerase can't bind to the promoter region and to begin transcription. So it makes sense that if you do not have lactose present, you don't want to waste any energy making lactase or enzymes that are going to break that down. So you want to be able to turn off and turn on genes depending upon what's involved. And so if that bacteria is functioning and growing in glucose, for example, it doesn't need that enzyme. If it's growing in lactose, then it needs that enzyme. Okay, so that repressor protein remains in place. Now look at that repressor protein. It's a three-dimensional structure with a piece that fits into it. And so when we add lactose to this medium, which the bacteria live in, it diffuses into the cell and then it attaches to that lac repressor. And when it attaches to the lac repressor, it binds to that lac repressor, then that becomes disengaged from that operon. And that allows the RNA polymerase, which is present, to translate this lactose utilization enzymes. And so there's actually three different proteins here that there are genes that produce that product. Okay. So lactose binding with that, and then when it binds with it, that protein then changes shape. All right. So that was prokaryotic cells. So let's take a look at eukaryotic cells. So eukaryotic cells and gene regulation, they have these TATA boxes, TATA boxes. And so it's either a sequence of TATA or TATA-AAA. It's a sequence of repeating nucleotides that code for gene regulation. All right, so now you want to take a moment and think, gee, what are they going to ask me in this test? They're going to ask me if it's regulating a prokaryotic gene or a eukaryotic gene. If it's a prokaryotic gene, you're looking for these operons and promoter regions in here, okay? And then for eukaryotics, you're looking at the TATA boxes, all right? So we have the same similar setup. We have transcription factors, which means those are things that are going to help transcription take place. And we have the DNA RNA polymerase, which is going to produce that copy of RNA off of the DNA. Okay, and here's our gene. So we have this box here. All right. So that, the site, we have these transcription factors that bind to the DNA at different places. And then the DNA goes in the direction of the transcription. Now, what you want to do is go back to yesterday's video again and take a look at that RNA polymerase. Actually, I'll stick just a copy of it into today's assignment so that it's there. But you're going to see that folding and that kicking. All right. So by binding up those DNA sequences in the regulatory regions of those eukaryotic genes, transcription factors control the expression of those genes. And so those transcription factors, again, are the things that are going to control gene expression. Transcription is what's going to happen. Factors are going to be controlling those. All right, so let's take a look at cell specialization. So if we're a complex gene regulation, eukaryotes is what makes differentiation and specialization possible. All of our cells have the exact same DNA. So we have to be able to express specific regions of those genes to make specific cells, have specific functions. And so the cells that make up the muscle cells in your heart are not going to have the same characteristics as the cells that are operating in our mouth, for example, making saliva, cheek cells and bone cells. All these cells that we have all have exactly the same information, but we have to be able to turn off and turn on different regions of that DNA to be able to get those cells functioning properly. Okay. So that is that regulation of gene expression. So gene expression means that that's what you're producing. And that's important in shaping how multicellular organisms developed, as I just explained. So each of our cell types found in the adult originates from the same fertilized egg cell. So we all come from one cell. Think about that, one cell. All right. So there's a bunch of research that's been done on homeotic genes and how they regulate organ development in fruit flies. And these things called homeobox genes code for transcription factors, and Hox genes determine the identities of each body segment. And so I'll elaborate on that a little more. Epigenetics is epi being outer, mechanisms control which genes are on and off during each stage of these insects' life cycles. So think about that these insects go through all those different life cycles. They have the same DNA, and something is controlling, which genes are turning on and which genes are turning off, and typically the environment that they're in, because they're not going to we don't have butterflies in the wintertime, right? Then there's environmental factors. So if you raise a rabbit, these Himalayan rabbits, in temperatures below 25 degrees C, they end up with these characteristic markers of black and white. If you raise those rabbits in temperatures above 30 C, those rabbits are white. So if you take a rabbit and you put an ice pack on the rabbit's back and raise that rabbit, and that ice pack is a cold place, it'll have a black patch on it. There's other influences, like with reptiles, for example, and the sex that the reptile will turn out has to do with its temperature, the temperature that the soil's at, coloration of moths, and other insects have to do with the time of the year that they're raised. And so there are many, we're a combination of both our genes and our environment. So, all right. Well, that will take care of that section. So it's about regulation.